Okay. I believe Dr. Kovacic is going to share his own slides. Okay. So do you need me to do anything up here? Nope, don't touch anything. Okay. Hi, Jason.
Hi, good morning, everybody.
Oh, it's late afternoon here, Jason. What time is it in Australia?
It's 5:40 in the morning on Sunday, so good to see everybody and good
So
to
the
be with friends.
so the next time you ever feel like doctors don't care about you, remember Dr. Wells, who drove 3 hours, Dr. Dix, who drove 2 hours, and Dr. Kovacic, who's up at 5:45, giving a talk from across the ocean in Australia. So grateful. Dr. Kovacic is probably one of the smartest people that I know, and one of the tallest people that I know as well. I think he might be the tallest member of the Society for Vascular Medicine, and I might be the shortest member of the Society for Vascular Medicine. So we get along great. He is the executive director and CEO of the Victor Chang Cardiac Research Institute in Australia. He serves as the Robert Graham Chair and Professor of Medicine at the University of New South Wales and is an adjunct professor at the University of Western Australia. He's a practicing cardiologist as well and a brilliant geneticist. I'm so grateful to Dr. Kovacic because I don't know that anybody, very few people can talk better on the genetics of FMD. Thank you, Jason.
Thank you so much. It's great to be with friends and doing this across the ocean. So it's my pleasure to speak to you today about the genetics of FMD. And I will just go back and just pick up on a comment that I heard a few minutes ago to say that FMD was first described in 1938. So we've known about this disease for decades, almost 100 years. And even in that very first description of the disease, it was understood that this was a non-inflammatory vascular disease. And very early on, it was appreciated that the disease likely involved changes in collagen and matrix, which is something I'll come to. So I'm going to dive right in with that as the starting point into the genetics of FMD and what have we learned. So this all started, and I can pick up from where Pam left off just before, that the first gene we ever found that was linked to FMD is this gene PRACTA1. And I'll come back to PRACTA1 a few times. It's a fascinating gene, and we've learned so much from this. So going back over 10 years to the first international FMD conference that Esther and Pam Mace, Heather Gornick organised in Cleveland, Nabila, who's a great colleague and friend and collaborator, came up to us at the time, and there were really no genes associated with FMD at that time, came to us and said, we've found this single gene, this single sort of genetic address that was borderline close to being associated with FMD. Not quite, but you can see it almost gets to that blue line there. And could we help validate this gene? So at the time, Nabila had assembled 259 cases with FMD. We had a couple of hundred. Some other people, Santhi Ganesh had some. There were some at Mayo. We put all of our DNA samples together, managed to come up with 1,154 cases, and managed to validate this very first gene associated with FMD. And I'm still I think we're all very proud of this paper that came from that international collaboration, published about 10 years ago now, linking the gene PRACTA1 to FMD. And that really started the whole ball rolling. And I think what was wonderful about this paper is, as Pam was talking about, it really set us up for international collaborations. And there's people on this paper from Europe, from Australia, from the States. It was a global effort, and this continues today in our collaborations. So having found just this one gene, PRACTA1, that was linked to FMD, it was a no-brainer to see if this same gene might be associated with SCAD. So we did a similar thing, some slightly different people, different names. David Adlam, you can see, is the first named author there from the UK, but we pulled all of our samples from SCAD DNA and found that exactly the same gene as was linked to FMD was linked to SCAD. And I think this mirrored what we were seeing clinically, that a lot of patients with SCAD had FMD and that the genetics were starting to come out somewhat similar. What is fascinating about this PRACTA1 gene, and we've thought about this a lot, we've been looking at it from many different ways, is that this PRACTA1 gene is not only linked to FMD and to SCAD, but other studies have linked it to cervical artery dissection, so carotid and vertebral artery dissection, to migraine headache, and also to hypertension. But that same gene is also linked to coronary artery disease, coronary artery calcification, and sort of hardening of the arteries. But it's linked in a paradoxical or opposing way, so that if you have one specific set of genes at this DNA address, you're more likely to have FMD or SCAD or cervical artery dissection, etc. But if you have a differing risk pattern at the same genetic address, you're more likely to get hardening of the arteries. And this genetic finding, I think, is really profound and important because in many ways, it is a bit of a silver lining to the whole story of FMD and SCAD. And what this is telling us is that if you have FMD or SCAD for that matter, you're actually genetically less likely to develop atherosclerosis, which is the regular hardening of the arteries that we talk about. So you're genetically somewhat protected from this. And this actually speaks to this sort of opposing relationship between these diseases, that they are not the same, they're actually quite different. And in many ways, you can think of a dissection as something that happens when the artery is a bit too soft. And the opposite problem, hardening of the arteries, is when there's plaque buildup, fatty plaque buildup, and the genetics of that are quite different and actually somewhat protective against having FMD and SCAD. I'm not going to go into this in much detail, but we've known about this gene for about a decade now. PRACTA1 turns out to be an extremely complex gene. We've been studying it for a long time. We've actually now developed mice where we've totally wiped out, we've edited out this gene. And you can see there, we're studying these mice that are deficient in the PRACTA1 gene to try and understand them. I would say to all of you, we use mice. It's really important that we use mice in our animals, in our studies on FMD, because without these animal models, it's really difficult to make any progress. But we're actually at the point of publishing some really important data. And I can share with you that these mice have some sex-specific findings. So there's some things that are changed in the female mice that are different in the male mice, which is, as you know, what we see with FMD. So following on from that discovery of PRACTA1, what we actually did then was to combine our samples from around the world. And we managed to actually get over 1,000 DNA samples, pulled things together and did what's called a genome-wide association study, which is just a fancy word for trying to find out what genes in total were linked to FMD. So we've increased the number of samples and we increased the number of genes we'd linked to FMD up to 5 genes. We validated that PRACTA1 was an important gene, but found another 4 genes linked to FMD, LRP1, LEMA1, ATP2B1, and SLC24A3. And if you've never heard of those, well, neither had we when we actually found that they were linked to FMD. This was all published about 4 years ago. And again, you can see the number of authors there. And I think it speaks to the great collaborative spirit that we have around the world. People from France, from Europe, from the States, from Australia, all collaborating together, combining samples to find these important results. I'll come back to those genes in a moment. But I wanted to speak about yet another study we've done on the genetics of FMD. And this was a study that we led out of Mount Sinai. It's still ongoing with Jeff Olin, myself, Daniela, Katie, and Dodoff. And it took advantage of the fact that fibromuscular dysplasia was named that for a reason. And it's because of this fibrosis that we can see in some of the arteries and this likening to fibroblast cells. And even going back, and I don't know if Pam, you'd found this paper when you were looking at the literature, but even going back to the 70s, there were studies done showing that there's changes in these fibroblast cells in the arteries, in the artery walls. And this is a special type of cell, but it turns out that it's quite easy to get these fibroblast cells from taking a piece of skin, and just a small piece of skin biopsy, we can derive fibroblast cells. So over a decade ago at Mount Sinai with Jeff Olin, we initiated what we call the DEFINE study. We initially set out to just enroll 200 patients. It was initially funded purely by philanthropy, and we started back in 2012. And as I said, the design of this study leveraged the fact that we can get fibroblasts from a piece of skin. This is the upper inner arm of a patient, and this is a 2 mm punch biopsy, same thing a dermatologist uses to take a biopsy, a mole or a freckle. And we can take a piece of skin. You can see there's these 2-3 mm pieces of skin there in that liquid. We can then take them into an incubator and grow those pieces of skin out in special techniques. And what we get after about 6 weeks is these pieces of skin that are here, and this is a piece of skin in the bottom right corner, and these thousands of fibroblast cells which grow out over the plastic plate. And we're then able to harvest these cells, and from those cells we can obtain the genes in them, RNA, and then look at those genes. And there's a technique called RNA sequencing, and enables us to look at the levels of up to 100,000 genes in these cells. And just a shout out to Valentina de Escamarad from my lab, still works with me in Mount Sinai in New York. She's done an amazing job running this study. The study is still ongoing. I said we'd set out to get 200 subjects. We're now up to about 450. And the design has been so powerful that we've continued to recruit and continue to enroll people into this study. So it's about half FMD cases and about half healthy controls that don't have FMD, don't have a family history of any vascular problems. So one of the things we wanted to do with this study was to understand the interactions or the gene networks that occur to cause FMD. And you can think about this as a sort of domino effect, or biologically, if you have one genetic change, that affects adjacent genes and causes other effects. Biology is complex, right? And so it's not just one gene in isolation. There's actually multiple genes acting together. And it turns out that these genes, we can sort of think about them as almost a pyramid of genes stacked up like this. And whilst there may be 1 or 2 genes at the top, they can affect hundreds, even thousands of genes that are downstream of them to cause the biological effects of FMD. And these effects are complex, as you know. So one of the things we set out to do was try to find these genes that are right at the top of the apex of that pyramid that are controlling everything that goes on down below to cause the features of FMD. So I'm not going to really explain much of this, but just to say there's a complex biological program that we enacted to do this with RNA sequencing, DNA sequencing, building these networks, validating these networks, looking at mouse models and things to try and understand this. But at the end of it all, we identified this network of genes, and you can see it depicted there, that was closely associated with fibromuscular dysplasia. This particular network comprised about 775 genes, and the top 14 apex genes are shown there. And for various reasons, we chose to focus on this one gene, UBR4, that appeared particularly relevant to FMD. Now, why do we choose UBR4? Well, one of the things we found about UBR4 is that it's highly present. There's high levels of this gene and protein present in the vascular wall. And you can see here we're looking in the internal mammary artery, in the renal artery. That red color is UBR4 that we're seeing in the wall of the artery. And it was present to a large extent, mostly in smooth muscle cells and in fibroblasts. Leapfrog ahead about 3 years, we found that we were interested in this UBR4 gene. We actually then created a mouse model where we deleted the gene or knocked out this gene, removed the gene UBR4 from smooth muscle cells. And you can see there, normal mice, there's lots of red. And where we've removed the UBR4 gene, there's a lot less of it present in the wall of the mice. There's still some cells that are in the middle of the artery, the lining cells, they're called endothelial cells, but it's mostly missing from the smooth muscle cells. And what did these mice look like? Well, they're alive, but you can see there immediately the aorta of these mice was about 40 to 50% larger than their healthy control mice. And that's in the abdominal aorta. This is in the thorax. That's actually the aorta coming off the heart, going right up into the neck. These aortas were about 40 to 50% larger than in the control mice. These mice didn't have a string of beads, but we never really thought the mice would develop a string of beads because the biology of the mice is so different to humans. The mice have a resting heart rate of around 500 beats a minute. Blood pressure, heart rate, all the forces that are acting on the vessels are so very, very different to humans. We never thought we'd see the same things. You can see here, looking under the microscope, we again saw these vessels were much bigger, more stretched out, and their size was larger. So interestingly, it was actually Esther that just a year or so before we were studying these mice had shown that arterial dilation is a feature of FMD. And Esther and her group, Brian Wells, published this paper showing that the aorta is actually larger in patients with FMD. So in the end, this feature of the mice that we found was actually reminiscent of some of the features of FMD that had only recently been appreciated. So when we looked at what is this network of genes doing, like some of the genes that we'd already found with FMD, PRACTA1 and some of those other genes I showed you earlier, it was pointing us towards changes in collagen matrix. Now it's collagen and matrix that are responsible for the integrity of our skin. And as we get wrinkling, as we all do as we get older, that wrinkling comes about because of changes in collagen and matrix that are affecting our skin. And it turns out that the genetics of FMD in this network were mostly about changes in collagen and matrix. This is just some of our scientific data looking at changes in collagen and matrix levels when we reduce the levels of UBR4. And you can see there are a whole bunch of collagens there. Col5A2, that's CO5A2, Col1A1, Col4A2, Col3A1, and right down the bottom, fibrillin-1. Fibrillin is actually the protein that is involved in Marfan's. As you know, Marfan's, we get dilated aortas. That's not to say people with FMD have Marfan's. Absolutely not. But these all of these collagen and matrix proteins and genes appear to be somewhat altered, slightly altered, leading to FMD. So just to summarize this particular network study, we found this whole network that's strongly linked to FMD. We found that UBR4 can control this network. When we take out this UBR4 gene, we knock it out, we recreate some of the features of FMD, and these enlarged arteries in mice were mostly because of changes in collagen and matrix. So we were really thrilled. This was published about 9 months ago in one of the Nature family journals, and they actually put this on the cover of this Nature journal, Understanding Fibromuscular Dysplasia. You can see there's a sort of kind of weird picture they put there, half a mouse head, half a human head, interconnected by this network. We didn't draw that, but anyway, it was a thrill to see FMD on the cover of a Nature journal. So just to summarize, what do we know about the genetics of FMD? I would actually challenge anyone that anymore says we don't understand FMD or it's a mysterious disease. We do understand a lot about FMD now. It's complex, I would definitely say that, and definitely it's hard to understand, but we do understand the genetics of FMD, and I don't think it's mysterious anywhere near as much as it used to be. We know that this PRACTA1 gene is a fascinating gene. It's associated not just with FMD, but also with SCAD and coronary artery disease. With this international collaboration, we've found 5 genes in total linked to FMD. We've now found this trade-off in risk between having FMD or SCAD and hardening of the arteries or build-up of fatty plaques. Through this network approach with fibroblasts, we've found this whole gene network with UBR4, and we've found that many of the genes linked to FMD cause changes in collagen and matrix. So I'll just end by acknowledging all the people, the funding bodies that have supported us along the way. Lots of great collaborators, including Esther and Pam, Heather Gornick. And just to mention what Pam just did, we were very fortunate to get the great news a couple of months ago that we were successful in receiving funding from the Leduc Foundation, which is an international organization that has funded us to the amount of US 8.5 million for this international network to better understand the genetics of FMD. And a shout out to Santhi Ganesh, Nabila in Paris, Stephanie Debet in France, Mark Lindsay in Boston, and Manuel Mayer in London that are the collaborators that will work with me to try and even better understand the genetics of FMD going forward. So thanks very much. It was a pleasure to be able to give this presentation.